My name is Lars Humblestone, and thank you to the Flavell Memorial for funding my research. Microbacterium tuberculosis is the causative bacteria behind the disease tuberculosis and currently infects 1.7 billion people worldwide. Despite this, rates within the developed world remain fairly low. We owe much of this success to the use of effective antibiotics. However, in recent years, we have seen the rise of drug-resistant strains of tuberculosis. To combat the increasing levels of resistance, scientists and doctors have been using whole genome sequencing. This involves taking a sample from a sick patient, culturing it, isolating the tuberculosis DNA, and then sequencing it to produce an output. However, this data is still quite rough and is not particularly useful. Firstly, we have to trim the data to remove artifacts from the sequencing. We then align the sequences against a reference genome before performing further processing. We can then compare our data back to the reference genome to get a list of mutations present from the sample. However, this is still not particularly useful. We can compare our data to that of a mutation database, thus giving us a list of mutations that we know cause resistance. Finally, we will be able to translate this data into an easily understandable clinical form so that doctors will be able to prescribe the correct drugs for the best patient outcome.